about the effect of the 21 gamma benzylidine digoxin 21-BD on cell addition and cell migration. Leandro, please come there.
Thank you. So I'd like to thank you for the invitation. It's a pleasure to stay in a cell biology meet again. And of course, stay in Cancun for a few days. I was good with friends. So I know that the talk is kind of different from the other sessions, but we're going to talk about sodium potassium base. And in my perspective, we're going to talk about new development of drugs that can interact with the pump and have some effect in cell adhesion and cell migration. So I'm going to talk beginning with an overview about the pump and the membrane for you can follow the next speakers. Okay? So just to make a introduction, the sodium pump. The sodium potassium is a ubiquitous enzyme expressed in all cells because you need the function of the pump for transporting the ions. So it's transported free sodium from outside the cells and pump 2 potassiums from inside the cells. And this is very important because the cells use this electrogenic force to couple with several functions. So the resting potential, you need the activity of the pump for that, action potential as well, co-transport of several amino acids, sugars, neurotransmitters. You need the sodium glycolates for use this, and as well the counter-transporter for proton and calcium. And because you have more sodium outside the cells, for doing this transport of sodium, you need the hydrolysis of ATP. So you have to spend the energy for that. So this is probably the most iconic figure of the pump. We're gonna show, probably Gustavo is gonna show this again for you guys. So the pump, you have some subunits that needed for the function of the pump on the membrane. So the alpha subunit has 10 transmembrane domain. It's the catalytic subunit of the pump, so and it makes the transport of the ions. So when you saw about the transport, you're always going to follow for the alpha subunit. It's very important for the function. The other subunit is the beta. Beta is very important. It's only one transmembrane domain. It's very glycosylated, and it's very important for the assembly of the membrane, the protein for the membrane. So we always found the protein is a lateral diagonal with equimolar amount of alpha and beta in the membrane. And the beta one is going to be like you're going to see in the other talk. It's very important because it's considered an adhesion protein. And for some tissues, we have one additional subunit that we call gamma, but it's a broad family that is the best is FXYD proteins. That is important in that it can modulate the kinetic properties of the alpha. So depending on the tissues, this is going to be a very huge impact in how the pump is going to make the pump function. So besides these subunits, I have several isoforms of the pump. So in the alpha I have alpha 1, 2, 3, and 4. Alpha 1, I said, is ubiquitous. Every cell has. If you don't, alpha 1 not work, the cell will not survive. Alpha 2 is more specific for heart, brain, and muscle. Alpha 3 is much important for the brain. It's much important for the brain. You have several diseases that have a mutation in alpha-3, you're gonna have problems in developing the brain. And alpha-4 is a very unique of the isoform because you only find this alpha-4 in testes in baby spermatozoids. So Gustavo talk is going to be about the specific functions of alpha-4. The beta-1, we have beta-1 that has all over the cells. Beta-2, beta-3 having depends, but we don't know much about how the isoforms of beta works. And as I said, you have the FXYT family that has 7 isoforms. So the FXYT2 that you found largely in kidney is the most important FXYT associated with the sodium pump. Another thing that is interesting is because you have in the structure of the alpha subunits, you have one very interesting difference between rodents and humans. If you see this arginine in the aspartic acid here, that in humans you have glutamine in the asparagine. So this difference in the rodent alpha one decreases the sensitivity for the ligands of the alpha subunit that was cardiotonic steroids. I'm going to talk in the next slide. So this is very interesting because considering all isoforms of the woman is sensitive for those drugs, in the rodent you have this less sensitivity. So this is important for models of study. So one important, very established pharmacophoric group that binds to the sodium potassium ATPase is cardiotonic steroids. Was probably one of the old pharmacophoric groups discovered in the 18th century for really Italy. Digoxin was the first one, and in several countries still is using for congestive heart disease. So you have, if you see how the structure, chemical structure, have the steroid group core with a lactone that is very important for the activity. And you have glycoside, sucralose, that depends, is very important for the function, for the activity, the biological activity in the sodium. So that's why if you have a glycoside, sometimes you can see digoxins are digitalis because it's coming from digitalis planata, or you can see that it's a cardiac glycosides. Right now, it's more often say cardiotonic steroids because some compounds that are derivative from frogs, like bufalin, they don't have the sugar. So, as an old drug, is how it works. So, digoxin is binding to the pump and it blocks the pump. So the pump, after it binds to digoxin, will not be able to transport any more the ions, so it can increase the concentration of sodium, internal sodium. This internal sodium concentration can inhibit the sodium-calcium exchanger. And because of that, it can increase the calcium inside the cells. So this, for some purpose, is good, because if you increase the calcium, you can increase the contraction. So that's why it's used for congestive heart disease. But if this effect was so strong, you're going to kill the cells. So this is the major problem with digoxin. It has a narrow therapeutic index because depending of the dose can have the benefic effect or can kill the cells. But something that is very interesting in our field is that we are working with the sodium potassium ATPase always as a pump, always as a transport through the membrane. And maybe like 30 years ago, Dr. Jansky and Dr. Amir Asghari from the University of Toledo came with this new idea that maybe the sodium potassium base is not only a pump, maybe it can work as a hormone receptor. So they demonstrated several papers in the literature that the alpha-1 subunit isoform can act as a scaffold protein, especially in the cavella, and can be associated with several signaling pathways proteins like SARC, PI3 kinase, AKT, ARC, EGFR. And if you change the concentration, the amount of alpha-1, you can trigger these effects. Or if you bind something for the alpha-1, you can change the conformation, you can release this protein for starting triggering the activity. And they show that one of the ligands that is important for this is cardiotomic steroids. So in the literature, what you have very well established, it's warfarin. It's worked very well to trigger these effects. And after that, we open new perspective for using the pump, not only a transporter, into kinetic experiments, but see what is the importance of the alpha-1 in several cell biology pathways, and perhaps try to modulate the effect with the cardiotomic steroids. So, what's the problem that you have? Let me again. So you have warfarin, you have digoxin, but again, it's narrow therapeutic index, very narrow, very cytotoxic. So, can it be possible to make some modifications of warfarin or digoxin to improve these effects? This is, in the synthetic, chemistry synthetic point, I feel is very, very hard, because this steroid group is very difficult for them to need some modification, chemical modification. So when you saw in the literature, the approach that people use is take out the sugar and make several substituents here, the sugar part. Or maybe, depending on the compounds, you try to see, take these hydroxyl groups, try to make some addition there. But even though it's very difficult, and we always have one concern about the lactone ring, because we know that the lactone ring is very important for the activity. So people don't want to change this lactone ring, afraid that it have anything. So in my lab, we try to do the contrast. So I'm a very lucky person, because I work very close to a synthetic chemistry. And the guy, he gave, look, it's going to be difficult for you to change the molecule, but perhaps you can find something that's going to be easier and cheaper for you. And he found one synthesis that you can add aldehyde in the lactone ring. Very quickly, one step, very reliable for you. And we try to do not using warfarin, that was the compound that's most used in the literature. We try to use the digoxin. Why? Because the digoxin is already used in the clinical trial. It's already used in clinic in several countries. So we believe that if you want to develop a drug, probably a digoxin derivative is going to be more easier for you go into the market. So after doing this, we're starting to check how going to be this effect on the sodium potassium base. So you can see here that comparing with digoxin, look the inhibition. Digoxin has very, very potent inhibit the pump, but the 2-NBT didn't cause the inhibition. Only high concentrations inhibit that has tissue that has more expression. And we're wondering if if this sees the effect in high concentration, but it's be able to bind the pump. So we do some experiments with the displacement of a radio-labeled warfarin. You can see that in high concentration, you can displace the warfarin, so it binds the pump. So it was important. 2-NBT is able to bind the pump, but only directly inhibited with high concentration. So this was the first change that this modification in lactone ring was from digoxin. Another thing that was surprise for us is like, when we started to doing this, our focus was for cancer. So we have several papers showing that the vaccine had effect in cancer cells, organ as well. And we said, look, let's try to make some modifications. We're going to find some cytotoxic effect. And it go on from that. But for my surprise, the IC50 is 50 micromolar. So in perspective for cancer biology is horrible. In comparing with digoxin, you can see here, HeLa cells like only high concentrations in kill the cells. So this is not good. But the science is wonderful because you close one door, we are upset because we want to work with cancer. But at the same time, this was a very good opportunity because if this compound shows some biological effects, we cannot be, we know the biggest problem to work with cardiotonic steroids, that was the cytotoxicity. So people not using digoxin because of the narrow therapeutic index. So this problem is solved here, essentially. So we started to see like, okay, let's see if we're gonna have some effect in some orders, cell biology effect that we have already shown for the WABA in digoxin. So that's why you move on to see the adhesion. So it's well known that WABA can improve the cell adhesion, and if you put too much cardiotonic steroid, you're going to detach the cells. The cells are going to lose this adhesion. So we started to see first is how it's going to be in cells, in living cells, how the compound is going to react. And it's very interesting. Even that it's not inhibit the pump, 10 micromolar of VT1BD was able to increase the levels of activity of the pump. And the RNA of the alpha-1 and beta-1 isoforms. So we started to believe, okay, beta-1 is a protein of adhesion. So maybe it can be useful to increase its adhesion in cells. And we started to check how the compound is going to find the action in tight junctions. So, incredibly, you can see that you have a huge increase in transepithelial resistance that show the functionality of the tight junctions. So after like 24 hours, 36 hours, you see that you have increased very, very consistently of that. And this is related with the increase of collagen 4 and Z1, that is constituent proteins of tight junctions. But collagen 2 is down-regulated for that. And you can see here, there in the immunofluorescence. So the treatment with 1BD have the reinforce of the collagen 4, down-regulated collagen 2, and Z1 very, very strong. So this for us was important. So, okay, I have this effect. It's not related to the cell death, but related to reinforce the junctional complex. But our doubts still remain. These effects is really through the pump, because it's binding not so well in the pump. So for check that, we started to use our model of MCK cells that is sensitive for the cardiotonic steroids, into one model that is resistant to cardiotonic steroids. And as you can see, the same effect we found in the increase of the transepithelial resistance, but in the resistance, we don't see the effect. It's the opposite. And for the adherence proteins, you can see here clear. The sensitive, looking how much is the expression, increased expression of the beta-1 and the beta-catenin, protein, it is totally localized along the membrane. And when you see the resistant CK cells, you don't see this effect very clearly. So it's nice, this effect, that the modulation of the increase of the adherence proteins is through the pump. And, okay, so if you have more protein of the junctional complex, probably you have more adhesion of the cell contact will be more strong. So we perform a calcium switch experiment. When you treat the cells with AGT, you remove the calcium from the media, so these proteins that was in contact between the cells losing the contact. So the cells open. And then when we add the new media of calcium, you can see that the media that has warfarin or 20-MBD, the epithelial resistance is increasing quickly compared with the control cells. So this shows for us that yes, we have an up-regulation of those proteins, in those proteins up-regulated for 20-MBD is causing more adhesion stronger on those cells, right? And you can see here clearly there when you follow this same effect with the fluorescence for the proteins of tight junctions. So you see here after you put the new media, of course, the control, but if you have the 21-MBD treatment, look how much Claudin 2 and Claudin 4 you have forming that you have these proteins already in the membrane making the contact. Another thing that's interesting, but you will never go further, is one protein that's very important for the assembly of these tight junctions is STAT3. Look how much you have the STAT3 expression compared with the control. So 20-MBD probably has signaling pathways for modulate this. We didn't have time for show all the data, but I have already data showing that it can modulate for EGFR, SARC, and other proteins. So of course, if this protein, if this drug can reinforce the adhesion, probably they're gonna impact the migration of the cells. So in this concentrated 50 micromolar, the sensitive cells, we blocked the migration, and the resistant cells did not. So again, it's depend of the pump. But until now, we just follow the protocol to work with high concentration of tranquility. Because you always think about cancer cells, maybe not killer cells, but maybe the cancer cells can inhibit the migration. But it's showing that warfarin has some biphasic effects. If you have one effect with high concentration, but low concentration, you can find some different effects. So it will try to see like how I can use this information on adhesion and immigration in a different perspective for selling the drug better, to be that maybe can be important. So we started to think about, okay, let's test this drug in very low concentration to see how this drug can deal in maybe in repair wounds. That's important for the cells migration, adhesion, everything. So again, using mouse fibroblasts, you see no change, no effect, cytotoxic effect with 21-MBD. The migration was not affected because we wanted to, like in a perspective of repair, that the migration going to be increased. But we didn't see the fibroblasts, these effects, either with warfarin or low concentrations of 20-MBD. But very interesting, when you do the stress fiber with colloidin, you do not see many changes in stress fiber, but you can see that the fibroblasts is have a conformational change in the fibroblasts. They are more elongated, they are more sharp. So we believe that this one kind of activation is compatible with some activation of the fibroblasts. So you have to measure, of course, how it's going to be the production of collagen. But I think that it's a very good indicator that maybe it not affect the migration of the fibroblasts, but probably can affect the functionality of the fibroblasts. But another cell type that's important for the wounds is the keratinocytes. So we're testing the human keratinocytes just to compare, look the MDT assay compared with digoxin, very cytotoxic. Here don't have no effect. And interesting for the keratinocytes, we saw this effect of the migration. So when you use 1 nanomolar, we can see that the wound that you perform is closed fast. So didn't have this effect for the fibroblasts, but for the keratinocytes it has. So this is very good for our purpose to repair the wounds. And when I saw this stress fiber, you can see clear, like the cortical ring of the actin is way more expressed compared to the control cells. So this clear show that probably if the cells migrate more, probably going to have more stress fibers because we need to occupy space, right? So this shows that you have this activation of the keratinocytes too. So we believe that probably this effect together with the fibroblastic activity, the keratinocyte activity, in low concentrations, it's going to be important for development of some products for wound healings. So just in summary about 20 is this gamma benzylidene modification is different for other catabolic steroids. So it's completely changed the behavior of the compounds, especially because 20-MBD was not cytotoxic, so this is very good thing. 20-MBD induces functional complex proteins, and those effects dependent on the sodium-potassium-base interaction. Inhibit cell migration in high dose, but activates keratinocytes in fibroblasts, and induce migration of keratinocytes in low concentration. So we believe that we need to do more experiments, of course, but I think that we are in a way for try to do and apply this in chronic wounds that's going to be very important for healthcare. So I want to thank all my collaborators. Sayonara was my PhD student that stayed in Gerardo lab for 1 year and a half. He did all this work in the tight chain proteins and the proteins. Carol was the master student that is doing the work in the epithelial cells, the keratinocytes, the fibroblasts. Jose Villar is the chemistry guy, so he's wonderful. Like it's not often common you ask for a synthetic chemistry that only wants to do different molecules. It goes in his office, look, I need 30 grams of the compounds. And he does this. So this is very important for us. Andrew thanks Ruben, and the staff, Gustavo Blanco, and Kansas City, was very long collaborators, and all people from my lab. Thank you.
Is there any question? Have you tried to stain integrins? Because you clearly show that there is an increase in stress fibers, and that suggests changes in the adhesion to the substrate.
Yes, it's a very good question. You never check it because you just move on for this new idea that's how the cells are going to be attached to the substrate. So I think it's going to be very important to see not either how it's going to be the interaction of integrins, the basal membrane, but probably see how it's going to be some modifications in the matrix intracellular. Probably going to be a lot of, we hope there's going to be a lot of suppression of proteins. It's going to be important for the remodeling of the tissues for repair.
Leandro, 23 fibroblasts are human fibroblasts?
Yes, right now, because when I start, that was the only available. But we start with the experiments with the human fibroblasts, because it probably going to be important because mouse fibroblast has the problem about the affinity.
And 33 are mouse.
Yes.
Okay. And another thing, they do not make tight junctions and adhesions.
See. Yes.
So maybe the difference that you see between fibroblasts and keratinocytes is because these are fibroblasts.
Exactly.
These are epithelial cells.
Yes, yes.
So in an adhesion context, it can be something.
I totally agree with you. It's so different. They all have their own role in the progression of the wound. So we have to be very careful to see what is the specific it does for the fibroblasts and what the specific it does for the keratinocytes. So probably reinforcing the adherent junctions. This is something that's interesting because in the first way, if you have more stronger the adherent junctions, probably this is going to be not good for the migration into the repair of the tissues. So using in a low concentration maybe can help not have this so strong tissue protein. But after you starting to fill the gap of the wounds, probably if it still has some effect on the adherent protein, it's going to be important because it's probably going to be easier for the cells making contact with that repaired tissues. So probably going to be like, you have to see how is the timeline of losing migration and then strong of the adherent protein to be more strong adhesion in this heal the wound.
I see. And do you remember the work from my lab when we add guava into CHO cells, which are fibroblasts? And what we observed is that there is an increase in ATPase synthesis, probably, we didn't check this, but arrival to the membrane. Now, a lot of ATPase in the membrane after treating the fibroblast with the guava. So maybe this is a point that we have to see what is the signal transduction.
Yes. This is another thing. Like if you saw our results of 10 micromolar, that you show more alpha 1 on the membrane, it's more beta 1 on the membrane. So one of our ideas is like maybe it's not really regulation of directly from the various proteins, but perhaps if you bring more pump to the membrane, you can assemble better the proteins of the junctional complex together. And this can reinforce all the junctional complex.
This is our idea because beta-subunit,
Yes,
as I will tell you immediately, is an adhesion molecule. So.
Exactly.
Thank you.
Very, very nice talk, Leandro. So, as you know, when you're thinking that there is movement of the sodium pump to the plasma membrane in other isoforms of the sodium pump, that's done through post-translational mechanisms like phosphorylation. Do you know if binding of your compound leads to phosphorylation of the sodium pump or some post-translational modification?
No, this is something that's important. To never check the phosphorylation of the pump using, but something that is in our radar. One thing that I can know that maybe is involved in all these mechanisms is because you have already data of 21D causal remodeling of the cholesterol in the membrane. So the increase of cholesterol has increase of phospholipids. And of course, this has a lot of the interaction with the pump, depends on how it's the microdomain of the membrane. You're going to have a huge impact in the activation of the pump. And perhaps how these extracellular matrix proteins can be interaction with those transmembrane proteins as well. So maybe one thing that could explain is how can it be the remodeling of the membrane.
Can I ask another question? So do you know if you have tried different cells with your compound? Do you know if caveolae are needed for your compound to act?
we publish a paper. This is interesting. So we publish a paper that uses HeLa cells that already has all the data about, but you're using K-culture cells. K-culture cells has interference proteins, but they don't have caveolin. And the effect was different. The effect that we found in HeLa cells, we didn't found one of them to have the same effect in K-culture cells. So maybe this is an indicator that the caveolin has some role, the signaling has some role. So we try to do knockout of the MCP cells to see if you found caveolin, how it's going to be the effect, what was another. So it's something that you want to try. Same cells, knockout caveolin, no caveolin to see how it's going to be.
Thank you very much, Leandro. So I am delighted to present now to Dr. Leora Shoshani from the Department of Physiology of the Center of Research and Advanced Studies from Mexico. She is going to talk to us today about new insights on cell-cell addition mediated by AMOC beta 2, subunit of the sodium potassium ATPase.
Buenas tardes. Hoy les voy a platicar de una isoforma de la subunidad beta de la sodium potassium ATPase, que es el centro de atención del laboratorio por los últimos como 30 años. Leandro nos dio una buena introducción de la sodium potassium ATPase y su trabajo se centra principalmente en la alfa, pero como les dijo, esta enzima o este transportador no funciona si no se ensamblan la alfa y la beta, las subunidades alfa y beta. Creo que se me brincó una, a ver. Okay. Entonces, yo me incorporé al Simvestab por ahí de 1995 al grupo del doctor Cerregido, y en ese entonces se sabía que la sodium potassium ATPase tiene dos subunidades, alfa y beta, y que cada una de ellas tiene varias isoformas. De la isoforma beta 2, que es de la que les voy a platicar hoy, se sabía muy poco, pero el principal trabajo fue del grupo de, necesito el pintador, no sé cómo lo voy a hacer, necesito otra mano, por favor. El grupo de Melita Schaffner en 87 publicó un trabajo que decía que, o más bien que identificaba una molécula de interacción, de adhesión en la glia. Le llamaban AMOC por sus iniciales, y en ese trabajo ellos muestran que el anticuerpo generado contra esa proteína inhibe, por un lado, la migración de células neuronales del cerebelo, y como podrán ver, sea un anticuerpo monoclonal o policlonal a distintas concentraciones, inhibía la migración, y usan otros anticuerpos como controles. Y por otro lado, ensayan la adhesión de neuronas a astrocitos. Igual, usan anticuerpos monoclonales y policlonales contra esa proteína que acaban de aislar, y ven que hay una inhibición específica de 25-26% de la adhesión de las neuronas a los astrocitos, pero no de astrocitos a astrocitos. Es decir, ellos detectan una proteína en astrocitos que promueve la adhesión de los astrocitos a neuronas. Sin embargo, los astrocitos entre ellos no se adhieren mediante esa proteína. ¿Por qué? Porque el anticuerpo que usan no lo inhibe. Entonces, la conclusión de ese trabajo era que AMOG es una molécula de adhesión heterofílica que debe tener un receptor en la membrana de las neuronas. Y lo heterofílico lo sugieren en base a que los astrocitos no hacen interacciones entre ellas. Voy a ver cómo la ingenio para que pueda usar todo a la vez. Entonces se publicaron varios trabajos con esa proteína después. Por ejemplo, este trabajo del 88, en el que incorporan a la proteína que aíslan de los astrocitos en liposomas y ponen a adherir los liposomas con la proteína fluorescente, más bien los liposomas fluorescentes, y ven cómo se adhieren a las neuronas. Sin embargo, si usan un anticuerpo en el medio, no se adhieren. Entonces, de nuevo, muestran que un anticuerpo específico contra esa proteína inhibe la adhesión de la proteína AMOG a neuritas y a neuronas. Entonces, deciden clonar esa proteína y ver de qué se trata. En 1990, Glur y sus colaboradores, igual del grupo de Melita Schaffner, clonan esa proteína, la secuencian y se encuentran con la sorpresa que es homóloga a la subunidad beta de la sodio potasio ATPasa. Y desde entonces, bueno, ellos muestran también que es la misma proteína AMOG y la beta ATPasa para concluir. Ellos confirman con ese trabajo que AMOG es realmente la subunidad beta 2 de la bomba de sodio y que esa es una molécula bifuncional que por un lado es importante para el transporte de sodio y potasio porque muestran que la guavaína inhibe su actividad, y también es una molécula de adhesión heterofílica que está involucrada en la migración neuronal y en el crecimiento de neuritas durante el desarrollo del cerebro. Entonces, desde aquí en adelante se le llama AMOG, que es Adhesion Molecule of Gear, Beta 2 Supplement. Entonces, cuando diga yo AMOG o Beta 2, es la misma, ¿ok? Una vez que ya se clonó, ya era la era de que la gente podía transfectar células y expresar proteínas de interés, clonaron la célula y la beta 2 y lo expresaron en fibroblastos L, que son fibroblastos de latón. Aquí vemos dos imágenes de microscopía donde están las células no transfectadas y transfectadas por la beta 2, y esas la usan como sustrato para crecer neuronas y miden qué tanto induce la interacción con las células transfectadas y no transfectadas del crecimiento de neuritas, y encuentran que a las 6 horas hay un aumento significativo en el crecimiento de neuritas o el largo de las neuritas, que va disminuyendo a las 10 horas y al parecer tiempos más largos ya no se nota la diferencia, pero a tiempos cortos sí se nota. También miden de distintas clonas que han generado, y bueno, la conclusión es que efectivamente también en la célula, no solo en los sistemas in vitro, sino en sistemas in vivo, células que expresan en su membrana a la beta 2 promueven el crecimiento de neuritas. Y interesantemente, en ese mismo trabajo, en el abstract, donde realmente resumen lo que se ha visto en este trabajo, ellos dicen que hablando en español, mándame Dios. Pues antes de que ponga, estoy hablando en español. Where is it switch? Okay, I will switch to English because nobody was complaining about my Spanish. Okay. They in the abstract, they summarize the differences in functional properties for the different beta isoforms of the sodium potassium ATPase suggest that among beta 2 but not beta 1 is able to interact with an unknown neuronal receptor leading to increased neural output, etc. I remark here the unknown neuronal receptor. So in 1993, it was clear that there are two beta subunits, beta 1 and beta 2, that beta 2 is an adhesion molecule in the glia, and that there must be an unknown receptor on the neuronal cells that bind to this molecule. And in the discussion, we suggest that the beta 1 subunit isoform may subserve other functions different from those of among beta 2 in the brain or other tissue. And in this moment of the history of science, I was introduced to the group of Marcelino Senefido in the senior staff. And I was introduced to the sodium potassium ATPase. I haven't worked with it before. And we knew that there are two subunits, alpha and beta. And in 1995, Gerardo Contreras and the group of Senefido published a work about a novel type of cell-cell cooperation between epithelial cells. And in this work, they described, or they had an observation that we could not explain at this moment. They saw that when they stain epithelial MCK cells with an antibody to the sodium potassium ATPase, it would be alpha or beta subunit. The pump was localized in the lateral membrane, as you can see in the image in the illustration, in the contact of cell-cell in the lateral membrane. When they used EGTA to separate the calcium-dependent adhesion, they can see that each of the cells bury its sodium pump in the membrane, which means that this line that we see here is composed actually of the pump of two neighboring cells. But the strange thing that they observe is that if they use a co-culture with other epithelial cells, MDCK cells will not express or polarize the pump in the membrane that contact this other cell. So this was very strange. And the hypothesis was that the beta 1 subunit is also an adhesion molecule that required the sodium pump polarization in epithelial cells. And this hypothesis was based obviously on the beta 2 observation as a molecular adhesion, and on the fact that when two cells, epithelial cells, are in contact, they do not express the pump in the polarized membrane. So what would be the strategic way to answer this or to test this hypothesis? We decided to transfect the other cell with the same beta subunit of the sodium pump of the MCK cells, which is the dog pump, and to see whether now, expressing the same beta subunit, it would be in the lateral membrane. And with Luisa and the group of Senefido, we made various experiments that were published in this work. And essentially what we saw is that while CHO wild type do not make it aggregate. When we transfected the CHO fibroblasts with the beta-1 subunit, they make big aggregates, the same as when we transfected with beta-gel, which means that these cells are now adhesive. And this is the quantification of this cell. And the important thing was, it is illustrated in this figure, when we mix or co-culture CHO fibroblasts with NBCK cells, we observe what I told you before, that the NBCK cells do not express the sodium pump in the membrane that contacts the other cell line that here is in red because we stated beforehand to distinguish them. And when I transfected the CHO, the beta-1 subunit of the dog cells, both NBCK cells and the CHO cells transfected express everywhere the pump, inclusive in the heterotypic contact. So indeed, we managed to show that the polarized expression of the pump in the cells depends on the association between the beta subunits located on neighboring cells. But actually this was just a suggestion because in this moment we did not do any adhesion assay. So the next question was, do they interact directly? And Teresita Padilla, which made a lot of protein-protein interaction assays using constructs that she made with green fluorescent and yellow fluorescent proteins. And she showed by pull down, immunoprecipitation, and FRET, that indeed there is a direct interaction between beta subunits. So in this moment of the history, around 2010, it was already established by our works and other groups that in epithelia, the sodium pump is established in the lateral membrane between neighboring cells, thanks to the adhesion of the beta subunit of the pump. And now we wanted to know what is the mechanism for beta-1 interaction. And in this moment, 2 crystals were available in the literature on 2007 and 2009. They published a low resolution and high resolution structure, crystal. And here we could have information about the beta subunit structure. And this enables us to do more precise questions about protein-protein interaction mechanism. Omar Paez, in his PhD project thesis, showed by using in silico methods that 2 beta-1 subunits interact and we localize the interface of this interaction. And we designed mutations in this interface in which he changed 3 amino acids to alanine in sequence along this interface, expressed them in CHO cells, and then assayed for adhesion. And we found that mutation 2, 3, and 4 affect the adhesion of the cells, while the mutation 1 on the adhesion. So this is why we suggest in the title of his work that the extracellular residues 221 to 229 in the Ig-like domain of this beta subunit is the the interface for this interaction. So now we have more clear idea about how beta-1 subunits interact, and the question was, what about the other beta subunits of this family? So we are going back to the beta subunits. We had this moment the structure based on the crystal of beta-1, but beta-2 and beta-3 till today are not crystallized, but there are methods, silicon, to model the structure of these subunits in base of the beta-1 structure. And now we are ready to study with more detail the mechanism of interaction of beta-2 in the brain. And this is what occupies us those days. What do we know about the beta-2 subunit in the brain? First, we know that the alpha and beta subunit distribution is tissue-specific. So in kidney there is almost exclusively alpha-1 and beta-1. In the brain, we can find 3 alpha subunits and 2 beta subunits. What makes it more complicated, because it came out that different cells in the brain express different combination of alpha and beta subunits. So in this work, they compare the expression in cerebellar cortex, in granular layers, cells, for example. We can find alpha-1, beta-1, and beta-2, while in astrocytes, alpha-2 and beta-2 are expressed. Here they have a question sign, but in the white matter of the cerebellum, astrocytes express clearly alpha-2 and beta-2. And in recent work from 2021, comparing the messenger RNA distribution, again we see that astrocytes express exclusively or alpha-2 and beta-2, although it is very important to say that the combination of alpha-1 and beta-1 is a housekeeping combination, so all the cells practically express this isoform. But the main isoform in astrocytes are alpha-2 and beta-2, and in this illustration taken from a review of Larson, they describe the distribution of the isoforms in different zones of the neurons and astrocytes. In 2019, Ethan and co-workers published a work in which they describe a functional interaction between the beta subunit of the sodium pump and myelin, which regulates growth factor signaling in cerebellar granule cells. And in this work, they present Western plots that show clearly the expression in microsomes and in granular cells of the 3 alpha subunits, 1, 2, 3, and the 2 beta subunits, beta-1 and beta-2. So it comes that there are neurons in the brain that express beta-2, while the dogma was that just astrocytes in the brain express beta-2. And this is our proposal based on the work we've done, that this interaction that they see in the granular cells is probably based on beta-beta interaction between astrocytes and granular cells, but this interaction should be homogeneous. And Luisa and Marlette, with Luisa and Marlette, we published recently a work where we show that beta-2 subunit is indeed a homophilic adhesion molecule. In this case, we expressed the beta-2 subunit in CHO cells. And first, we did an analysis in silico, where we show that two beta-2 subunits can make a dimer in a transposition. We analyzed the interface, which amino acids participate in this interaction, and Luisa realized addition assays with these CHO cells expressing beta-2 with the yellow fluorescent protein. And again, we observed very clear that these cells make large aggregates in a similar way that beta-1 do. And when we did the protein-protein interaction in vivo and in vitro, we found that these proteins, two beta subunits that we distinguish with different tags, can elute from a nickel interaction assay. And now we are ready to study the heterotypic interaction between beta-2 subunit and the unknown receptor in neurons. Christian, the PhD student in the lab, looked at VO3 and looked for reported interactions for beta-2 subunit. You know that nowadays everybody does, not everybody, but there is a lot of information about interactomics, so you can find data in the literature, and this is VO3 is a very useful web page. We entered the gene for beta-2 subunit, and we found that there are 70 molecules that would interact with the beta-2 subunit. So we selectively screened them, and we found that actually there are very little proteins that would be the receptor for the ATP1B2, the alpha subunit, which is obvious, but there was reticulon 4 and tetraspanin 31. These two proteins are from ER, and we are looking for plasma membrane interacting proteins, so we stayed with tetraspanin and reticulon. And Christian started to work or test if tetraspanin 31 is the neural receptor for AMOR22. This protein is part of a family of tetraspanins that it was identified in neocortex. There is no crystal or any 3D structure reported for this member of the family, and we have no information about tetraspanin 31 function in the brain. We don't know what it does there. But in general, tetraspanin's function as cis dimers. So we modeled the structure of tetraspanin dimer and docked it with the beta subunit, now with beta-2 isoform, the extracellular domain, simulated the molecular dynamics, and found which are the residues in the interface. And now we also transfected fibroblasts with this tetraspanin 31 tagged with the green fluorescent proteins, and this is the transient transfection, and this is one of the images we got while cloning these cells, and hopefully, and also by Western blot detects the proteins that are expressed, and what we are what we're going to do is some protein-protein interactions between tetraspanin 31 and beta-2 to test this possible interaction. So to sum up, among beta-2 is an additional molecule in the central nervous system. It can establish both homophilic and heterophilic interactions, and its expression is related with cancer, glioblastoma, and genetic disease, which I will talk today. And we are willing to perform protein-protein interaction assays that will test in vitro and in vivo hypothesis. So just to remind you that cell-cell interactions in the central nervous system are very important for the function of the brain, and we believe they are more better to participate in these interactions and probably will have significance in disease like cancer and degenerative disease in the brain. Thank you.
Questions?
I'm sorry for the Spanish in the beginning, but nobody made any sign. Tell me if I
You have another nice presentation, but one question. You showed some of the experimental results that using tunicamicin, there is no joint, no? So that means that glycosylation of the proteins are involved and you have not only complex interaction.
Yes, thanks for asking, because of time I didn't talk about it, but beta-2 subunit has 7 sites of N-glycosylation in comparison to the beta-1 that has just 3. And the group of Rita Schaffner and other groups have shown that the sugars are very important in neuronal cell adhesion. So yes, the beta-2 subunit is indeed highly glycosylated, and we think that the sugars take place or participate in this protein-protein interaction. Actually, we have a new working publication, which is in silico work, where we show that which of the sugars, of the 7 sugars, is in the interface and how it is affected this addition, we simulate it. So yes, the sugars are very important.
Do you think that the molecular structures that are involved in beta homocytic binding are the same that in the heterotypical binding?
No, I don't think so. So what we have done by now is comparing the beta-1 and beta-2 dimers. And they are not the same. They involve different amino acids, residues, and interfaces. And with the beta-2 and tetraspanin, exactly what I showed quickly because you signed it, there is no time. Here, what Christian is analyzing now is exactly this, which amino acids and residues participate here in the interface. So I hope we can answer it.
Thank you very much, Mira.
Who is going to talk to us about the testis, the specifics of the potassium yes, upon specifically the sperm function.
Well, thank you very much for the introduction. I would like to really give a huge thanks to Dr. Contreras for putting up this session and inviting me for this session. And also to Dr. Luis Felipe Jimenez and Dr. Lourdes Segura, because this is a wonderful meeting, a wonderful venue, and it's really a pleasure to be here. So I will continue talking about sodium, potassium, ATPase, but I'm going to focus on the newest or latest isoform found and its very specific role. So thanks to Leandro and Liora for giving a good intro. I'm not going to say, but that's the sodium pump with in gray it's alpha subunit, the beta, maintaining the uneven sodium distribution across the plasma membrane by hydrolyzing ATP. And here you have the different isoforms, 4 alpha isoforms for the alpha subunit, 3 for the beta. And one thing that is very interesting on these isoforms is that they are expressed in a very tissue-specific manner, and they have different biochemical properties. And as you can see here, alpha-1 is expressed with beta-1 almost everywhere. Alpha-2 is mainly expressed in skeletal muscle, adipocytes, as Liora was mentioning. Alpha-3 is in the nervous system. But look at this, alpha-4 is only expressed in the testes. And it's actually one of the most, in terms of its structure and amino acid sequence, most divergent sodium, potassium, ATPase alpha isoforms. So years ago, a laboratory in Cincinnati of Dr. Lingrel and us cloned these isoforms from a testis library. But it wasn't known if it was really a sodium, potassium, ATPase or other type of ATPase. And at that moment, we were working with this system, the vacuum virus system, which is based on the expression of proteins in insect cells. And we found out that SF9 cells, it's a clone of insect cells, do not express endogenous sodium, potassium, ATPase. And that has been the problem to study the properties of the different isoforms, because every animal cell expresses minimally the alpha-1, beta-1 isoform. So this system was very useful, and as you can see here, we, after cloning, express alpha-4 in the presence of beta-1. We made an antibody, and you see the expression in the cells, and these are the cells expressing alpha-4 and beta-1, and these cells are very good. They are able to deliver the proteins to the right membrane domain, the plasma membrane, and after infection. And as you see here, uninfected cells show no barrier. So with this, we had the possibility of studying the activity, and as you can see here, this is ATPase activity sensitive to wabain. And as Leandro explained, wabain is a specific inhibitor of the sodium, potassium, ATPase. And when you have sodium, potassium, and magnesium in the media, you have activity. But if you get rid of these ions, you don't. And then we did some other characterization studies, but definitely this is a wabain-sensitive sodium, potassium, and magnesium-dependent ATPase or sodium, potassium, ATPase. And that's how we knew that this was a new isoform, functional isoform of the sodium pump. And with our antibodies, we just screened a lot of tissues, and we found that it's only in the testes. It's abundant in sperm, and as you can see, alpha-1 is everywhere, including the testes and sperm. We have more recently done RT-PCR and a battery of different tissues, and it's only in the testes. So testes also express alpha-1, but there is no alpha-2 or alpha-3. And here, with our antibodies, the testis is constituted by tubules, the seminiferous tubules. And this is a section of a seminiferous tubule showing that alpha-1 is basically expressed in all cells. But alpha-4 instead is in the inner part of the tubule and a little bit in other areas. So we have done studies with GFP labeling too, and what we know now is that alpha-4 is expressed in the male germ cells of the testes, and it's expressed later after meiosis of the cells, and it's very abundant in the spermatozoa. And the spermatozoa are in the lumen of the tubule, and that's where they are released when they are differentiated. So if we isolate sperm, and we can isolate sperm from the epididymis, which is the structure that continues after the testes, and that's where the sperm matures and is stored until ejaculation. We isolated it from different species, but this is a mouse, and you can see that sperm also express alpha-1 like every cell, but they have alpha-4 and not alpha-2 or alpha-3. So, immunocytochemical studies show that this is for rat and human, and blue is DAPI, that's the nucleus of the sperm. Alpha-1 is expressed all along the sperm flagellum, while alpha-4 is also in the flagellum, a little bit in the head, but very, very little. It's mainly a flagellar protein, but it's expressed in this region of the flagellum continues here. It's mainly expressing what is called the mid-piece of the sperm flagellum. And we still don't understand, and in humans it's kind of the same. We don't understand exactly, we're studying this, why it's expressed there and not along the flagellum. So, also spermatozoa express the beta subunit, obviously. As Diora said, without the beta subunit, you don't have a functional sodium potassium ATPase. So beta 1 and beta 3 are present there, not beta 2, and they are expressed all along the cell flagellum here. And the interesting thing is, if you do pull-down assays, and using anti-beta 1 or anti-beta 3 antibody, alpha-4 comes along. If you do the opposite, immunoprecipitation using anti-alpha-4 antibodies, beta 1 and beta 3 show there. And the differences on the bands here is because of the different glycosylation that these proteins have. So alpha-4 then is able to assemble with both beta 1 and beta 3 to form two types of sodium potassium ATPases. We normally talk about isoforms when we call the different alphas and betas, but when they assemble, then they can assemble in a kind of promiscuous way, forming different isoforms of this sodium potassium ATPase. We have studied both alpha-1, alpha-4 beta 1 and alpha-4 beta 3, and they are very, very similar in their biochemical properties. So, but the big question is, okay, these isoforms in the testes, is it doing something different? So the ultimate experiment to see if there is any functional relevance of this molecular heterogeneity of the sodium pump is to just knock it out. And we knock it out in mice, and as you see here, this is the expression of alpha-4 in wild-type animals. In heterozygous animals, you get some expression, and in the null animals, you don't see any. Alpha-1 is constant in all three, and that's tubulin used as a control. When we look at these animals, they are viable, they are happy, they run and they do everything, but there is one thing that is very interesting. They can mate with females, but they are completely infertile. And it's only the males that are infertile, not the females. So why are they infertile? Well, we look at the testes, we look at the seminiferous tubules here, everything looks normal. These mice make sperm, they produce sperm, but when you use these this sperm to fertilize eggs in vitro fertilization assays, while the wild type, 24 hours later, form the two-cell embryo, the null mice are not fertilized. Heterozygous mice are functional, and with some expression of alpha-4, they are fertile. So something is very wrong with the sperm, right? Because it cannot fertilize the eggs. So what is wrong? Well, one of the big things that sperm do to fertilize the egg is to swim, right? And try to find the egg. So we studied, the first thing we studied was sperm motility. And we can do that using what is called computer sperm analysis, or CASA. And you can measure total motility or different parameters of sperm motility. This is like swimmers in the Olympic Games, right? The sperm moves in different ways, so you can study the curvilinear, you know, it's like backstroke or you know, free swimming, and you can study all these movements. So in green here you have the movement of the wild type, and in the red in the null mice. And as you can see, all parameters of sperm motility are very, very decreased. And this is just to show you movies here. That's the wild type sperm, happy, swimming, and being very, very nice. But when you look at the null mice, they swim a lot less, and look at those. Oh, that sperm is suffering there. It's not going anywhere. And on top of that, I will show you later, they have this bend. You see it has a bend in the flagellum. So the sperm is very damaged, obviously. Without swimming, they are not going to reach the egg. But the sperm undergoes a process called sperm capacitation. And that happens in the female genital tract. And that allows sperm to move more and actually move in this kind of frantic way once it finds the egg to get into the egg. Well, and this is called hyperactive motility. And when we measure hyperactive motility, as you can see here, the null mice basically have none. So, first of all, this sperm cannot reach the egg, and second, there is almost no way it can penetrate the egg. So the sperm, so alpha-4 then is very important for male fertility, right? So we did a series of studies to look at the mechanisms by which this is happening. And if you measure intracellular sodium in sperm from null mice, it's higher than the wild type mice. And that makes sense, right? If you don't have a sodium pump taking sodium out of the cell, it will accumulate in the cell. Well, when sodium accumulates in the cell, and if you measure the membrane potential, remember sodium potassium ATPase maintains membrane potential. The membrane potential of a wild type sperm is around -40 to -50 millivolts. Well, look at the null mice, it's depolarized. When you don't have a good membrane potential, those lose their capacity to be excitable, and then they can obviously not swim well. How does the sodium pump do this? The sodium pump contributes to membrane potential, but it doesn't generate it per se. You need potassium channels. So somehow there is a link between alpha-4 and potassium channels in the sperm to maintain membrane potential. The other thing is we studied intracellular pH, and we found that the cells are acidic compared to wild-type cells. And the reason for that is what Leandro was showing in his slide, and is that the sodium gradient that the sodium pump generates is important for the secondary movement of other ions in the cell. So, for example, the sperm contains a sodium-hydrogen exchanger. Sodium comes in, protons come out. So when you do not have alpha-4, the sodium gradient is dissipated, and now protons accumulate in the cell, and that's why the pH is reduced. And something similar happens with calcium. There is a sodium-calcium exchanger in the sperm as well as in other cells. So when you lose the sodium gradient, now calcium accumulates in the cell, and that's what we found that this sperm have. So all these parameters are crucial for sperm to swim. So those what we believe are the reasons why the sperm is not swimming. But then I show you too that if you collect sperm from the testes or the epididymals in wild-type animals, you see the head and the long flagellum very nice and straight. When we collect it from the knockout mice, you see that the sperm has this bend, and this has different degrees of angularity, to the point that sometimes it's completely retroflex in itself, as you can see here. So when we can quantify that. Wild-type mice in green have a lot of normal sperm. Few of them are bent, but look at the bent sperm here. It's much, much higher in the knockout. And the reason for that, we believe, is because there is accumulation of water in these cells. There is sodium retention, there is water retention, and sperm, different from other cells, have very little cytoplasm. At the moment water accumulates, the flagellum cannot modify properly to that volume, and it bends. And we have done scanning electron microscopy to look at the bend, and you can see here the bulging of the plasma membrane and the accumulation of fluid underneath. And sometimes, eventually, it will completely break, and the microtubular structure of the flagellum, called the axonema or axoneme, it just comes out. Here is electron microscopy showing a totally retroflex sperm with, part of the midpiece and then part of what is called the rest of the flagellum or principal piece bent together and with the membrane bulging all around. So this sperm is very defective. Well, one quick thing we did is you measure sperm motility. In wild type is high, in the null sperm is low. If we reintroduce alpha-4, we recover motility. We also recover hyperactive motility. We make the sperm also lose the bend, and the mice are again fertile. So alpha-4 is definitely important for the sperm function. So what we also decided to do is make another mouse model in which we overexpress alpha-4. And to distinguish this, we express in the mice the human alpha-4. And when you do that, this is the expression, and that was to distinguish it with a specific antibody. These are the 71st tubes showing human alpha-4 expression on top of the mouse. Here you have the sperm, alpha-4 expressed in the flagellum, the human form. And look at this. If we measure sodium potassium ATPase, this is wild type in green, this is the transgenic alpha-4 overexpressor. Total activity goes up. And the increasing activity is due to the alpha-4 that we are overexpressing, and alpha-1 is basically constant. So we have more alpha-4 expression. When we measure sperm motility, this is the wild type. The one expressing extra alpha-4 swims more. It has more motility. So somehow, and we don't understand exactly how this is happening, the sperm gains the capacity to move more. And maybe what is happening, we don't know this for sure, is that all the parameters that I showed you before, membrane potential, are improved. And then the sperm is like if you put more fuel in your tank, right? And you get a better combustion in your engine, you probably can propel the cells better, right? When we try to see fertility in vivo, it's obviously the same. Why? Because the limitation there is the female. I mean, there is only a certain amount of embryos that could be fertilized. But the fertility is the same between wild type and this super sperm that we have there. So let me show you some of these. I told you that the different isoforms have different biochemical properties. So something very good to study for the sodium pump is the kinetics. Remember, it's a pump, an ion transporter, but it's hydrolyzing ATP. so it also works as an enzyme, right? So if you can study the kinetics of this enzyme. Here you have activity of sodium potassium ATPase versus different increasing concentrations of sodium, potassium, and ATP. And all these are ligands that are important for activity. So as you increase them, the activity increases, okay? In different colors here, and this we did this in SF9 cells. We express alpha 1, 2, 3, and 4 in combination with beta 1 and study the kinetics of all these alpha beta pairs. And as you can see here, they are different, okay? And we could calculate the K1 halves for the different alpha and beta complexes. And as you can see here, alpha 4 has a high affinity for sodium, a lower K1 half, a low affinity for potassium, an intermediate affinity for ATP. And what is interesting is that if you measure activity in the presence of the inhibitor wabin, you get inhibition curves. Well, it turns out that alpha 4 is the one that has the highest affinity for wabin, and therefore is the one that is inhibited the most. So this is an interesting property here. When you express it in insect cells, this is it's a huge difference. Alpha 1 is relatively resistant, and the K1 half is in the micromolar range, for alpha 4 is in the nanomolar range. So when you look at sperm, basically those two curves are combined. Alpha 4 sensitive, alpha 1 resistant, because those are the two isoforms expressed there. And these are for rat and human, the K1 halves are similar. So, another approach that we had was like, okay, if you use wabin in different concentrations to only inhibit alpha 4 or to inhibit both, maybe we can mimic what happened with the knockout mice, right? So this is motility versus time. If you measure it in sperm, you get motility there. Let me get this started. The movie works. Yeah, there you go. But if you treat the cells with doses of wabin that only inhibit alpha 4, now look at this. The motility is much reduced, right? But if you use even more wabin to inhibit now not only alpha 4 but alpha 1, not much more happens. And this is the graph here. So what this means is that alpha 4 is the isoform of the sodium pump involving sperm motility. Alpha 1, it's not that important for this parameter, right? So with this idea now, knowing that alpha 4 is so important for male fertility and sperm function, we are trying to use this as a marker for male fertility and fertility, but also if we can inhibit it, we could make a male contraceptive, which is very needed and there's none. So what are we trying to do? We're trying to exploit this high affinity that alpha 4 has for wabin to use. We cannot use wabin because it's very toxic, right? That will be a good contraceptive, but it will eventually kill the poor guy, right? So we don't want to do that. So we are modifying the structure of wabin. And wabin, as Leandro showed you, has a steroidal backbone, a sugar, and then the aglycone or the lactone ring. So we have been making substitution in C17 and C3 here and made a series of derivatives. And more recently, we're making smaller molecules which are easier to synthesize. So let me show you some of the wabin derivatives that we have been trying. And the way we approach this was to express alpha 4 in the insect cells and then do inhibition curves, calculate the K1 half. And let me point out to three of our most potent wabin derivatives. And as you can see here, they have K1/2 in the nanomolar and actually picomolar range for some of them. So we have also been doing to guide the design of our compounds, homology modeling of alpha-4 to alpha-1 and docking studies, and we have docked our compounds and have been changing the structure accordingly to get compounds that combine better. And here I have to mention the role of my collaborator, Guta Gior, at the University of Minnesota, who is the one that has been making compounds. So at this moment, we have compounds that, as you can see here, inhibit alpha-4 with at very low concentrations. And these compounds not only are effective against alpha-4, but when you compare it to the other isoforms, we find isoform selectivity. So you need very high concentrations to inhibit the other isoforms, but with very little concentrations, you inhibit alpha-4. So that is looking good. Now we have selectivity. When we use, this is for one of the compounds, and we treat a sperm with these compounds, and look at the low concentrations here, relatively low, we can depolarize the cells, you can acidify the cells, or you can increase the calcium. Exactly the same mechanism that we obtain when you knock out alpha-4. So this is kind of proof that these compounds are working through the guanine binding pocket in the alpha-4 isoform. So what do they do with sperm motility? Well, these compounds inhibit motility. We don't get total inhibition of motility yet. You can see here, this is white, the control non-treated, and look, this sperm still moving some, but it's basically twitching. It's not going very far. So this is looking relatively good. All parameters of sperm motility are affected. And then we have been studying different things on the properties of the compounds. For example, reversibility. If we treat with the compounds and then we wash the cells and then keep measuring motility, the motility is inhibited, but it doesn't come back. At least for the time that we can maintain sperm alive and healthy on the CASA machine. Hypermotility is also inhibited, and the compounds also have a similar effect in human sperm. So we have done in vitro fertilization assays, and those you have to understand that these assays, they really maximize the possibility of sperm to fertilize an egg. You're basically putting sperm in contact with the egg, and we have a very important reduction in vitro fertilization after application of the compound, treating with the compound the cells. So then we wanted to move to in vivo studies, and we did some basic toxicity assays like anti-proliferative assays, showing that compared to guanine that will here basically make the cells not proliferate anymore. Our compounds do not interfere with cell proliferation. There is an assay called the hERG assay that is taken as a gold standard for toxicity of the heart, and these compounds are not toxic there. So with that in hand, what we decided to do is inject these compounds to either rats or mice. We have the similar results. We have been doing different protocols. In this case, we treated the rats with 5 milligrams per kilogram for different days. And then what we did is sacrifice the animals, take the epididymis, collect the sperm, and measure sperm motility. And as you can see here, the sperm still move a lot, but the motility is highly reduced, at least by 40%. So we also have used the compound at different doses, and we get a similar inhibition. So this is very important because after intraperitoneal injection of the compounds, the compounds can reach either the testes or the epididymis, bind to sperm, and the sperm that is collected later, has decreased motility. This is total motility. Down here you have progressive motility. So, because these woman derivatives are difficult to make, there's big molecules, we're working with smaller molecules. I'm showing you here one of the compounds that we call 116. It has it has isoform specificity, inhibits alpha-4, inhibits sperm motility. Let me see if I can get the movie going here. Right. Okay, there you go. See, so you see a lot less movement there than in the unkilling control. It's effective against human motility, is also reduced. And when we do in vivo studies, when in this case it's mice, you know, the compounds at different doses inhibit sperm motility. And of course, these are two compounds here, they are effective in vitro fertilization assays. So we're doing, I'm not going to spend too much time on this, but now we have been studying the permeability of these compounds to see how they reach the target. We are doing pharmacokinetics to measure the time that the compounds reside in plasma. And if you look here, the times for testes are longer. So, and we have collected also epididymis, which is similar. So the compounds are reaching their target. So we're not quite there yet, but we have compounds that show selectivity and may be good. So, just as a summary of the talk, I hope I didn't go too much over time, but alpha-4 is expressed in major cells of the testes after the meiotic phase, and it's very important for male fertility, for sperm motility, hyperactivation, and through the mechanisms that I told you, maintaining intracellular sodium levels, cell calcium, pH, and membrane potential. And the alpha-1 isoform cannot compensate for alpha-4, as you saw. So this is an isoform dedicated to sperm movement. The knockout is completely infertile, the complete knockout. And then I show you data that shows that it's an attractive target for contraception. So in working with WABA, we can exploit this property that the isoform has in binding WABA with compounds that can probably serve as eventually made contraceptives. And I would like to finish by acknowledging the people in my lab that at different times have done different things for the project. Mainly Jeff McDermott is a research associate in the lab. September Numada, student. Gladys Sanchez is another research associate in the lab. Jordan and more recently these other students are working in the project. This is my collaborator at University of Minnesota, Julia York, and this is from the urology clinic, AJ Nanja is an MD that helped us with obtain human sperm and other studies that we're doing in the clinic, and of course the funding by NIH. And I'll stop there. Again, many thanks for this invitation. Thank you for being here, and I'm open to questions now.
Very nice talk, thank you. The band in the sperm is in a specific site or it's random?
That's a very good point that you observe. It's always between the midpiece and the rest of the flagella. And the reason for that is that there have been studies using different drugs that by, for example, inhibiting potassium channels, water also accumulates. And the band is very similar. And the reason is that seems to be a fragile point in sperm. The mid piece is more structured. Mitochondria are there, the microtubules there. There is another structure called the outer dense fibers. But then the rest of the flagellum is more elastic. So when at the bridge between the two, there is a weak point. And then when water accumulates, that's why it bends there.
Okay, thank you.
Yeah. People have studied the mechanics and physics behind these for other purposes.
And actually, that's what I said, it's an osmotic problem. It's based on the literature that every time you osmotically challenge sperm, they start bending there.
Okay. And another point, your compounds inhibit around 50% or maybe, but not
The
totally.
compounds, you mean?
Yes,
Yeah.
the compounds, they inhibit just something, not totally. But as well as we know, for fertilization in human, you don't need 100%. When there is a small reduction in mobility, it's enough to get infertility. So your compounds are very promising.
That's a very good point. So, based for humans, and based in the World Health Organization, patients that have 40% motility or less are infertile, are cataloged as infertile. So we do not need to achieve zero, but it would be nice to be safe, right? But you're right. In reality, we do not need zero. The other important thing is that the compounds inhibit hyperactive motility. I show it, but I didn't stop, by 80%. And that is a very critical movement that the sperm has to penetrate the egg. So yeah, I think we are not quite there, but we're getting there.
Yes, nice talk. I'm an embryologist, and I was thinking about what is the role of this alpha-4 on the competence to blastocyst formation. Have you thought about doing an experiment using this kind of non-progressive null sperms by macro-injecting this sperm inside the oocyte and trying to see what is the competence of that cytox and what are the implications about implanting that kind of embryos? So what is the role of alpha-4 in the releasing, maybe, I don't know, the calcium intracellular levels during fertilization and competence to blastocyst formation?
Well, that's a huge question that is super relevant. In my institution, I belong to I'm a member of there is a reproductive center. And there is a researcher working on oocytes, and he had exactly the same comment that he had. And there are techniques that we have not done yet, but he's trying to help me to do this. Exactly what you said, there is a technique called ICSI, intracytoplasmic sperm injection. And when you do that, you know, and that's what it's used in the clinic. If you find a sperm that is not of good quality, you can, the surgeon retrieves it, even if it cannot be ejaculated, and then, you know, that can be injected directly in the egg. And yes, that's a way to fertilize. We have not done it yet, but our assumption is that the problems of this sperm are mainly in the flagellum. We have done tunnel assay and everything, and the nucleus seems okay. So if you inject the nucleus, I am predicting that this very damaged sperm can still fertilize it. Now, what happens with the offspring and all that? That's what this collaborator is trying to do. That's very, because yes, it's a problem, right? You may even achieve fertilization, but what is going to happen with, you know, the offspring, right? So,
As a conclusion, how did you do the null mutant?
how did
you do the null mutant?
That's a good point. Yeah, I didn't stop in the technology there. So what we did is basically flux. We use a CRISPR-Cas. We flux the alpha-4. If you remember the structure that Leandro showed, the alpha subunit has like 4 transmembrane domains, then it has this big intracellular loop between transmembrane 4 and 5, and then the rest. We flux all that medial portion, which is where the ATP binding site is the enzymatic core, let's say. And then when you use the creamice, you delete all that, and basically it also will cause a frame shift, so there is no expression of alpha 4. We completely deleted it. So yeah, but that's good point.
I have another question.
Okay.
When did alpha 4 appear in evolution?
So that's interesting. It appeared relatively later, and one thing I have to tell you, alpha 4, it's a mammalian form of the sodium pump. If you go back to other species, it's not there. And it's actually complicated depending on the species you look at. For example, if you go to fish, and the most common one, the Daniel, the zebrafish, there are like 9 isoforms there. So which one correlates with alpha 4? It's not clear. I haven't looked into that, but there was someone in my institution years ago that said, but then it was so complicated. Not even alpha 1 corresponds to alpha 1 in zebrafish. So it's difficult there. So my belief is that alpha 4 is important for hypermotility. So the sperm, I briefly told you that they undergo this process of capacitation. This is particularly a property of sperm from mammals. Sperm from other species do not, I mean some do, but and the capacitation process is not, it happens in the female genital tract. So basically, to explain it quick, males produce sperm, but the sperm is still not able to fertilize the egg. So from a women's perspective, you can say, my goodness, these males cannot make anything, right? So it needs a last process of maturation in the female genital tract, and that's called capacitation. And for that, there are other products, not only alpha 4, that contribute to that, and they are mammalian. In mammals, this is a property of mammals. And why the sperm need to undergo capacitation, it's not clear, but apparently it's an evolutionary selection that happened it's a very good example of coevolution. The female creates this environment, and the sperm has developed tools to undergo capacitation and fertilize the egg. But if you then put a sperm that cannot capacitate, it won't be able to fertilize the egg. So it's a selective mechanism that nature developed. That's what it's believed, right? Okay, I'm not, that's the best explanation that it's out there. Thank you again.
Thank you, Gustavo. As usual, wonderful talk. Now we move to the last talk. I'm going to present you our data on the role of sodium potassium ATPase in the control of tight junctions and other cellular functions. By this time, you already know that the sodium potassium ATPase bonds sodium and potassium that generates the typical concentration differences of animal cells with a high concentration of potassium inside the cell and sodium outside. You also know that the sodium potassium ATPase is inhibited by cardiotonic steroids, especially with bioavarin, which is the prototypical component of this family of substances. And Dr. Aliora explained us a new function that is addition on epithelial and glial cells that are provoked principally by the beta subunit with the participation of the beta subunit. And also, very interesting of this enzyme is another function. So, besides being an ion pump, it is an addition molecule and also a receptor that triggers intracellular signals to induce long-term change in different type of cells. I am interested in cell addition of epithelial cells, and I wanted to know if the sodium potassium ATPase and guanine can control cell addition in epithelial cells. So epithelial cells have this polarized transporting phenotype. Cells attach each other with a number of intercellular junctions, including tight junctions, adherent junctions, and also the sodium potassium ATPase expressed in the lateral membrane, and desmosomes, and they have junctions for the substrate, like the hemidesmosomes and the focal contacts. The plasma membrane of the sodium potassium of the epithelial cells is divided in three domains. The apical one, who is oriented against towards the light of the organ that the epithelia cover, and the lateral one, which is the one that attaches to the neighboring cell, and the basal domain, who attaches to the substrate. And when any particular substance can cross epithelia vectorially following two routes, the paracellular route, the transcellular route, using channels, pumps, and carriers in order to cross the cell. But substances have also the possibility to cross the epithelia in the intercellular space, passing through the tight junctions. And tight junctions control this transport very specifically, depending on the type of proteins of the tight junctions that are particularly expressed. So, when you culture these cells, in the absence of calcium, they lose contacts with the neighbors, but they attach to a substrate. If you recover the calcium in few hours, the adhesion between cells is going to be recovered. And in few minutes, you can see an initial formation of the tight junctions. Tight junctions are like a belt that surrounds all the perimeter of the cell. And when they form, they obliterate the intercellular space. So tight junctions are also delimit the frontier between the apical membrane and the lateral membrane. And they are very useful to keep the composition of each plasma membrane domain specific. So, few minutes after you add calcium, tight junctions start to be formed. And then some sodium potassium ATPases are trapped in the wrong side, the apical, because they are normally expressed in the lateral membrane. And the cell has a correcting mechanism to take those wrongly located ATPases from the apical membrane, and it is endocyte to be degraded. At the same time, new sodium-potassium ATPases that are storage in the interior of the cell are directly sent to the lateral membrane and are assembled and expressed there to perform their functions. So how does guanine detaches epithelial cells? Well, if you add enough concentration of guanine in the order of 300 nanomolar or more, one of the things that you are going to observe is that the cell detaches first from each other and then from the substrates. That observation leads us to ask if there is a connection between the sodium potassium ATPase and the expression of the proteins of the cell junctions, in general of the cell junctions. So we ask, how does guanine detaches epithelial cells? And we first question ourselves whether guanine control adhesion-related properties of the epithelial cells. Those adhesion-related proteins functions are very important. Those functions are migration, for example, differentiation, and first of all, we study what happened with cell junctions. At a high concentration that is enough to completely inhibit the sodium potassium ATPase and provoke cell detachment. And you can see here the normal aspect of a protein of the tight junction. They look like a honeycomb pattern. And that's because the protein is expressed only in the places in which one cell attaches to the other. And you can also see that guanine, 300 nanomolar, induces the endocytosis and degradation of tight junctions, in this case, clotting 2. But the same happens with other proteins of the tight junction, clotting 4, occludin. All of them are transmembrane proteins, but also CO1 is a protein of the tight junctions, but it's an associated protein of the membrane. You can block this endocytosis and also the degradation if you inhibit this protein, the small GPP binding protein CSAR with PP2 in all the cases, meaning that this detachment process, this endocytosis of the proteins of the tight junction and its degradation requires a signaling pathway in which CSAR is involved. And we also found that the MAP kinase ERK1/2 is equally involved in this change of reactions. When you measure by the monoclonal quantity of the proteins of the tight junctions, you see that the proteins decrease under the treatment with 300 nanomolar guanine, and you can block at least for CO1 and occludin and partially with clotting 4, but not with clotting 2. You can block the process with this substance that inhibits ERK1/2 activity, meaning that MAP kinase is involved in the process of detachment. The degradation of the proteins, of course, takes place in the lysosome. So this degradation is inhibited if we add baphyloisin, which is an inhibitor of the proton pump of the lysosome. So there is endocytosis, and there is degradation, lysosomal degradation, and that suggests that perhaps autophagy is involved. So in order to study if autophagy participates in the degradation of the proteins of the tight junction, we use this system, which is a plasmid that expresses the protein LC3 coupled to the red fluorescent protein and the green fluorescent protein. So in the regions of the cells, the autophagosomes, which are organelles involved in the process of autophagy, in the autophagosome, when these proteins are expressed, given that the pH inside the autophagosome is neutral, you see the expression of proteins, and therefore you see green line on the fluorescence, like is observed here. You see the red one, the green, and then the yellow as a combination of the two. But in autolysosomes, in which, because of the action of the proton pump of the lysosomes, the pH is very acidic. The green fluorescent protein, which is more sensitive, its fluorescence is more sensitive to changes in pH, to the acidification, it stops to fluoresce in those conditions. So you can see that with guanine, we still can observe the red signal corresponding to our LC3. LC3 is a crucial component of the machinery for autophagy. And we cannot see the green fluorescent proteins coupled to LC3, and therefore in the match only the red component is present. That demonstrates that guanine induces autophagy. We then move to try whether components of the tight junctions co-localize with autophagosomes. So we label autophagosomes with a component of this structure, which is the protein P62. And then we saw how much clotting 1, 2, or 4 co-localizes with this structure in intracellular regions that are known as puncta, and we know are lysosomes. And you can see here the analysis of the colocalization, and there is a high colocalization whenever guanine is present. As the process of degradation is taking place continuously, in these experiments it's very important to inhibit the degradation, so to increase the possibility to observe the localization. So these results, these increments in the correlation between the green and the red signal demonstrates that guanine is inducing autophagy, and that the autophagosomes have the components of the tight junction. So, another step to advance in the study of the signaling that leads to the degradation of the tight junction proteins, we resort to the study of the protein, which is a transcription factor very important for addition, which is the protein STAT3. And we wonder whether STAT3 is involved in this process of degradation of the clotting. As you can see here, Guanine induces the degradation of chlorine-2, using here the normal localization at the plasma membrane of this protein of the tight junctions. And here we see the expression of STAT3 in the cells. We measure the total content of this STAT3 protein and observe that when the cells are incubated with guanine, the total cell content of STAT3 decreases. Even more, STAT3 can be phosphorylated, and its phosphorylation is crucial to deliver this transcription factor into the nucleus. And we observed that the first event, even before the degradation starts, is the inactivation of STAT3, the dephosphorylation. So, in order to disassemble the tight junctions, STAT3 must be inactivated. So, we are now studying what is the role of STAT3 inside the nucleus, and those experiments are being realized now. So, then we ask ourselves, what happens if we use guanine concentrations that are more lower than the ones that I showed you until now. These concentrations is 10 nanomolar, and surprisingly, the effect on addition is just the opposite. When we culture epithelial cells with 10 nanomolar guanine for several days, what we observe is an increase on the signal of some of the proteins of the tight junctions, like chlorine-2 and chlorine-1, also including 4. Some others, like O-chlorine, do not change very much. And this increase in the expression of the protein in the region of the tight junctions is caused by an increase in the synthesis, in the amount of the protein that you can see here in those graphs. Each protein have a particular kinetic in order to reach an increase in the amount. O-chlorine does not. At this concentration, the cell keeps the potassium normal, so guanine is not affecting the ionic gradient of the cell. But nevertheless, is inducing an increase in the tight junctions, and of course, an increase in the function of the tight junctions. Tight junctions in the presence of 10 nanomolar guanine are more sealed than in the absence of this. And those changes, those increases in the sealing and in the change in the composition of the tight junctions are mediated by CSAR as well as PD and the MAP kinase. So, if we detach the cells using high concentration of guanines, we get the activation of the CSAR MAP kinase pathway. But we also have the activation of the same pathway when we use 10 nanomolar guanine, but in both cases, the physiological result is opposite. In the first one, with high concentrations, the cells detach, and finally, they are going to die. And in the second case, at low concentrations, tight junctions are going to be more sealed, and the epithelia has a normal structure, and the cells are going to survive. we are interested in study what difference in the activation of the cell and of the same pathway can lead to opposite results. Now, here I have a scheme in which it's useful to explain how the inhibition or the binding, the occupancy of the guanine in the sodium of potassium ATPase start signaling pathways that, depending on the concentration of guanine, can lead to a reinforcement of cell-cell adhesion or to the detachment and death of the cells. And they are mediated by the same signaling pathway. So, guanine controls adhesion, cell junctions. Then we move to study whether they control the differentiation of the epithelial cell. In order to do that, we choose the formation of the cilium. Every cell, including the epithelial cells, have a major cilium, which is like a sensor of the environment. We measure the development of the cilium in NPCK cells, and we found that cells treated with guanine have longer cilium. You can see here the statistical study showing the increase in the length of the cilium. And during the formation of the cilium, the first step is the formation of these structures that are seen as dots in the fluorescence and are called procilium. And you see here a completely developed cilium. If we count these structures in the cells culture in 10 nanomolar guanine as a function of time, what we can see is that while they follow the normal pattern in the control cells, in the presence of guanine, the formation of ciliated and procilated cells are accelerated. And this acceleration also depends on the same signaling pathway that I showed you in the previous results. So, in conclusion, guanine also controls the last stages of the polarization and therefore controls or induces the differentiation of the epithelial cells. You see here, there is a normal process of development of this cilium, but if you add 10 nanomolar guanine, you get the differentiated state days before than expected. So, another property that depends on cell adhesion is migration. We studied whether 10 nanomolar guanine induces any change in migration. So in those scratch assays, as you can see, is that guanine increases the degraded distance of cells, and that this process of motility is inhibited by the same drugs that I showed you before, that block CSAR and that block the MAP kinase. We did some other studies in which we measure the secretion of metalloproteinases based on the assumption that perhaps what guanine is doing is to increase the secretion of any of those proteins. And we found that metalloproteinase 2 is secreted in higher quantities in the cells that are treated with guanine. So, what guanine does is that it inhibits it binds to the sodium potassium ATPase transactivates through CSAR the EGF receptor pathway, activates therefore the MAP kinase, and then induce an acceleration of the migration of the cells to repair the wound. So in summary, one adhesion-related proteins in epithelial cells. Low concentrations induce the synthesis of globins and increase the sealing of tight junctions. High concentrations detach the cells, induce endocytosis and lysosomal degradation of tight junctions proteins, and also autophagy of those. One accelerates celiogenesis, implying that is increasing the differentiation of the epithelial phenotype, and it accelerates collective cell migrations. And all those processes are mediated by the same signaling pathway involving SARC, F12, and STAT3. And finally, I have to recognize the participation of all the people from the laboratory. This work was performed by all these people. This is, as usual, science is a work made by teams and not by a specific person. So we are ready for discussion. If you have any question, I'd be happy to answer it.
Yeah? I know you, obviously. But I have this problem with the two concentrations of the wabain and the opposite effects. So, what do you think? Why is this difference? Are they two different populations of ATPase in the membrane, or what happens?
You know, we work with the same cells in both cases. We use the same wabain. In fact, we use the same batch of the wabain, and you still get very different results depending on the concentration. What the hypothesis that I would like to try in the future is that the spatiotemporal signaling is the one that is important to define any differentiation state to state. You know that wabain at low concentrations in epithelial cells, Apelia's group has demonstrated, induces oscillations in the intracellular calcium concentration of renal cells. What I would like to see if there is some, these oscillations can change in frequency and amplitude, and that could lead to a very difficult result physiologically. So I would like to try just to use those new biosensors of CSAR and MAP kinase and see if there is any difference in the spatiotemporal activation of those kinases in the microscope with the fluorescent kinases.
Very nice talk. I had a question. You said that, wabain, as you showed very nice data, is increasing celiogenesis.
Go here because there isn't much reverberation and I understand.
So you mentioned that wabain showed nice data on inducing celiogenesis. Do you know if the cilia are normal?
Well, we don't know. Morphologically, they are normal. Just they reach the same length at the end. The only difference is the genetics. They do it faster than the control cells. And the electron microscopy images, we don't see any difference. The width of the cilium is the same. It's just the length which changes. The procilium is just similar. We have not observed any structural difference, although we are using very low resolution confocal microscope to see those. It would be those results. It would be very interesting to use high resolution microscopy to see if the cilium, the procilium, the initial stages of the cilium formation are different in both cases. But I don't think so. They look similar. It's just the genetics.
So I have another question. Is there a correlation between ciliogenesis? It looks like yes, right? And all the tight junctions and all these other proteins. And if so, what does that mean?
Well, there is a correlation in the sense that changes in the tight junction takes hours or 1 day, maybe 2 days. And changes in the cilia used to take longer. The formation of the cilia is the last stage in the formation of the polarized epithelial phenotype of transport. But you know, we can observe changes in the tight junctions before we detect changes in the cilium. So they have different time windows. So if there is no, I thought you were all going to ask. So if there are no more questions, I hope that after this symposium, you value more the sodium potassium ATPase. You know about the cardiac glycosides, who are very interesting substances that can be useful not only as poisons and weapons, they are also useful as medicines to cure maladies, and perhaps they are important in the physiology of the cell as well. And they can be of endogenous origin. This symposium is more related to cell physiology, and I hope that you have enjoyed it. And thank you very much.